There's a type of stem cell in the skin. There's a type of stem cell in muscle. The central nervous system has 3 types of stem cells. The liver has a particular type of stem cell. So there are single tissue focused types of stem cells. They haven't really been used that much in practice. And it's a bit controversial because it gets into the ethical realm. There is one company that has made tissue-derived stem cells, say neural tissue derived from fetuses, and that could potentially be used. And the question is, well, does it really work any better? Other types of stem cells and is free of the issues that are around that.
What if aging isn't about slowing down, but leveling up? I'm Dr. Isaac Jones. This is the Longevity Leaders Podcast. Welcome to the Longevity Leaders Podcast. I'm Dr. Isaac Jones, and this episode is part of our exclusive series recorded at the Exponential Longevity Summit, a global gathering of cutting-edge health experts exploring the edge of human performance, health span, and longevity. Let's get into it.
Welcome, everyone, to today's exciting session and episode. I am Dr. Rudy Mueller, and I am thrilled to introduce our next guest, someone truly at the forefront of regenerative medicine and longevity science. Joining us today is Dr. Todd Ovokaitys, a Johns Hopkins trained medical physician, accomplished author, researcher, and he was really pioneering new frontiers in health, regenerative medicine, and healing. And really, his groundbreaking work in regenerative medicine includes the development of the SONG laser technology, as well as the utilization of VSELs, which are very small embryonic-like stem cells. He's had multiple medical publications and a recent FDA trial approved. So I hope to dive in deep into these different categories and talk and expand upon the FDA trial, as well as where he sees this research going and the possibilities of regeneration and repair. So I'm honored to welcome Dr. Todd to our conversation today. Thank you so much for joining us. I know our audience is excited to dive into this incredible work. So I think to start, I'd like to hear a little bit more about what first inspired you to kind of explore this world of regenerative medicine and longevity, and then maybe dive into V cells and stem cells and all this good stuff.
Well, Dr. Rudy, a great pleasure to be with you. I'm excited to have this discussion as well, both areas that are kind of near and dear to our deepest areas of finding ways to create best outcomes. And there is a lot of art that goes into that, as you know. And I have coined the phrase, the biohack is the stack. 'Cause it's not just one thing, it's how you put all the pieces together and individualize them for a given circumstance. The interest in longevity really goes back to when I was about 13 years old, and I fell in love with DNA. DNA became my thing. I studied a whole lot about DNA that was way outside of any of my school curriculum. And when people ask me why I became a doctor, I'd say it was really to keep all avenues open for the study and exploration of anything and everything about DNA. Because even when I was 13 years old, I had the inspiration, the intuition that if we understood everything about DNA, we would be able to learn how to keep the body young for a very long time and probably how to reverse any adverse condition of the body as well. And that vision really hasn't changed since then.
That's amazing. At 13 years old, you started down this path.
Mm-hmm.
That's
Yeah.
great. So with V cells, I mean, if you don't mind just giving me a little bit of background and the listeners a background on, what are V cells, what are other types of stem cells, and, how do they differ?
Mm-hmm. It's an excellent question. And stem cells have become a hot topic. There's a lot of work being done in the area. There are lots of different types of stem cells used. And there have been many great results in various circumstances and applications. And I think the biggest challenge is that the word stem cells makes it sound like it's one thing, but it's like saying the word color. We can say, well, paint the walls a color. Well, which color and for what purpose? The understanding probably is best going back to the beginning and laying out the hierarchy of stem cells, because there's a whole system that begins at the beginning with the fertilized egg. And during the first few divisions, you have very powerful stem cells. And when you get to roughly the 30-cell stage, it's a very tiny group of cells called the blastocyst. And if you took those cells apart and put each of them in a womb that was ready for it, you could potentially make 30 whole new people. So those very early cells are true embryonic stem cells. And they're so powerful, they're called totipotent, which means each cell can make a whole new person. That's really powerful. And in the realm where the questions come up, it's the one type of stem cell that has a risk because it's so powerful that it can form tumors, typically benign tumors that are called teratomas. And they're really weird. They can be as big as a cantaloupe and covered with hair. So it can be a literal giant cantaloupe-sized hairball with skin and teeth and nails and muscle and bone and cartilage in a really weird tangled mess. They're generally benign, and if they get too big or encroach on something, might need to be removed. But that is a reason why the power of them might need to be mitigated by the potential risk, at least with that type of stem cell. And there were some limits placed on the study of embryonic stem cells. In part, there were some moral and ethical considerations that's beyond the scope of this discussion. Yet there was also a true medical consideration, which is they're so powerful that they can sometimes make weird masses of cells that don't really belong there. At least people generally don't want them there. Just past totipotent is pluripotent. Pluri means many, and potent means quite powerful. And a pluripotent stem cell is the next step in the hierarchy. A pluripotent stem cell can then go to make the three primary germ layers, which we probably call from biology the endoderm, ectoderm, and mesoderm. As a pluripotent stem cell, because it's at the tip of this chain and can make the three other germ layers, in essence, it has the capability to make any type of cell. A totipotent cell can do that, yet the challenge is you don't necessarily want it making clusters of types of cells you don't want. Whereas a pluripotent stem cell can make any type of cell, it also has various lines of evidence to verify that they don't have a tendency to teratoma formation. So they're very powerful, they can make any type of cell, and they appear to be safe from the complications that can occur with an embryonic stem cell. And that's where the VSELs come into the picture because they are a pluripotent stem cell.
Hmm.
And before we talk about the unique features of this previously controversial type of cell, I'll go on in the hierarchy. The three primary germ layers, while there are three, there's really one that has been focused upon and used a lot, which is the so-called mesoderm. There is a lot of work in the area of what are called mesenchymal stem cells, or MSCs. And these can be derived from fat. That's a common source. Adipose-derived MSCs are used quite a bit. They can also be derived from bone marrow. And in an area that's a little bit more of a gray zone, they also can be derived from umbilical cord blood. And we'll get into some more distinctions. Beyond mesenchymal stem cells, which are commonly used, there really hasn't been much use of so-called endodermal or ectodermal layers. The endodermal tends to make digestive structures. Most of the GI tract is endodermal. The ectodermal makes central nervous system tissue and skin and teeth and other things. Those are the big things that it makes. Those really haven't been particularly called upon. The level past the mesenchymal stem cells, which that layer and what they've been used especially well for is regenerating musculoskeletal structures. MSCs can make cartilage, bone, ligaments, tendons, and muscle. So MSCs, if used for that purpose, have had many good results. There are, beyond the germ layers, there are single tissue type stem cells. There's many of them. Pretty much every tissue of the body has its own type of stem cell. There's a type of stem cell in the skin. There's a type of stem cell in muscle. The central nervous system has 3 types of stem cells. The liver has a particular type of stem cell. So there are single tissue focused types of stem cells. They haven't really been used that much in practice. And it's a bit controversial because it gets into the ethical realm. There is one company that has made tissue-derived stem cells, say neural tissue derived from fetuses, and that could potentially be used. And the question is, well, does it really work any better? Other types of stem cells and is free of the issues that are around that. In theory, ultimately, in the stem cell world, there may well be neural-derived stem cells, muscle-derived stem cells, cartilage-derived stem cells that are designed to make a specific tissue. And there is some work done in that area, but I haven't seen it make it to practical application yet. There is a core distinction in the stem cell world, whether the cells come from the same person or from somebody else.
Mm-hmm.
If it comes from the same person, it's called autologous. Auto means self. And even the FDA has been fairly allowing with respect to the use of your own stem cells, because they come from you, they go back to you. There are guidelines around that. In general, they have to be used within 4 hours. And the key phrase is minimal manipulation, which means not adding other chemicals or growing them in culture, which might alter their characteristics. But autologous stem cells have been widely allowed if used under the particular guidelines. Allogeneic means it comes from somebody else. And probably the most common use of allogeneic stem cells is the use of umbilical cord blood. And those have been available. Sometimes people have to go to other countries to get them with the US restrictions, like Dominican Republic or Panama. There are some famous places doing that work there. And the biggest question with allogeneic stem cells is whether they actually will persist in tissue. So the use of allogeneic MSCs, for example, there is the understanding that they tend to remain in the body for about 6 weeks. And there can be a regenerative effect, particularly the cytokines and growth factors that they make. Yet whether they actually create that much new tissue that stays is another type of question. There may well be some, but it tends to be limited by how well the cells are actually matched to the person. When we get into allogeneic, there are questions of tissue compatibility. So if there is a major transplant, like a liver transplant or a kidney transplant, before doing the transplant, they need to actually match the tissue. And there's a type of testing called HLA testing, which is a mouthful phrase, human leukocyte antigens. And there is a panel of antigens, and these are proteins that the cells express once they differentiate. I could go deeper down the rabbit hole with the different classifications. Well, I will a little bit. There's MHC class 1, and those are expressed on all cells. And there's MHC class 2, which are only expressed on some cells, like lymphocytes. And basically, the HLA matching has to show a compatible match with both class 1 and class 2 antigens. I could go further into the distinctions, but that's probably enough for a general understanding. The most basic set of antigens would be looking at 6. There can be some further levels of refinement of matching, but the most basic would be matching 6 out of 6. And for a major organ, for compatibility, generally you would need to have a perfect 6 out of 6 match, or there would be a need for chronic immunosuppressive therapy so that the body doesn't literally just reject those cells outright. The use of allogeneic cells then gets into a whole different arena. With allogeneic cord blood, for example, the FDA allows the use of that blood from first-degree relatives. So if it's a brother or a sister or parent, that is allowed because there's going to be significant intrinsic compatibility because basically it's from the same genetic line. Technically, allogeneic cord blood in a non-first-degree relative, it's not technically allowed, and I will go further into saying who or where. I mean, in some cases, just for it to be fully allowed, that would be done in other countries that do allow it. The utility issue, if serendipitously there is a good enough match between donor and recipient, then those cells may well be incorporated as a person's own and regenerate new young tissue. And you really can't get much younger than umbilical cord blood. That is literally 1-day-old blood. Where the VSELs become very interesting, which is the short way of saying very small embryonic-like stem cells, or VSELs, which is the usual phrase as they're expressed in the literature. The VSELs are very early. They're literally at the tip of the differentiation chain, and they're pluripotent. And something very else unusual happens with them, which we might consider a type of gift from source, as it were, or at least a biological gift, that for unknown reasons, the V cells go into relative metabolic dormancy around the time that we're born. And because they're metabolically dormant, they're not dividing or divide infrequently, which means that the telomeres aren't shortening and they remain very young biologically. Also, because of the tip of the differentiation chain, when they're given person to person, it's their cells, it's the same genetic match, they're perfect fit, they're compatible. It is our belief that not only may they replace adult older cells with young robust cells, they may also reverse the age of the various stem cell pools of the body.
So that's the aspect there. It's really pluripotent in that it's going to impact multiple tissues, not just one, where you're talking about, like, we have liver stem cells and skin stem cells and, different types of nervous stem cells, few different types of nervous system stem cells. That pluripotency from the V cells and the bonus that they are biologically young and that they are autologous coming from yourself, you're going to have a better match and potentially better outcomes.
Right. Because they stay young, there is evidence that they may undergo a slow aging process, yet remain many years younger biologically than the person, that they are capable of literally going through the body and replacing older stem cells with younger stem cells. So you kind of get a double bonus. There is kind of a system-wide ability to take any adult cell population and replace them with cells that are biologically younger that have more replications left in them. That could be another part of the discussion. And uh, also replace the stem cell pool that is regenerating that tissue with younger cells. So there's an immediate rejuvenation effect and a longer term rejuvenation effect. I mentioned how many cell divisions they have. You've probably heard of what's called the Hayflick limit.
Yes.
And to define that, if you start with a cell that's just been created, say a new lung cell, the question is, how many times can that cell divide to replace cells that are lost before it becomes senescent, before it stops dividing, before it becomes a senescent cell? And you've probably had the discussion about senescent cells that they're alive, but they become inflammatory and they tend to actually accelerate the aging and inflammatory tissue effects around them. So the Hayflick limit, as I recall, for pneumocytes, for lung cells, is about 60. That seems like a lot, but a lifetime is a long time. And when I had my early discussions about V cells, one of the extraordinary pieces of information was that they got to about 200 and they stopped counting. So the V cells may have an exceptional biological potential for many cycles of replication, yet retain their biological stability. So another reason that they would be useful for regenerative effects. It's also incredibly useful that they're very small. One of the issues with mesenchymal stem cells, if they're used outside of the realm, for example, of injecting them into a joint space to repair cartilage, ligaments, and tendons, that they're relatively large. The typical diameter of a mesenchymal stem cell is 12 to 30 microns, which is pretty small. However, the diameter of a V cell in the dormant state is really small. It's truly very small, as they say, it's 1 to 2 microns. If you look at them under a microscope, they basically look like bare nuclei, which is about 1 micron, with a little bit of cytoplasm. And if you infuse MSCs or V cells IV, the first place they go to is the right side of the heart and then to the pulmonary circulation. And I like to quiz people, but I'll spare you that. I'll just tell you the average diameter of a pulmonary capillary is 6 microns. So it's just mechanics. It is not that easy to get a 12 to 30 micron ball through a 6 micron tube. And some of them can, get really tight and squeezy and squeeze their way through. Yet evidence I've seen suggests that on the order of 95% of the MSCs literally just get to the lung. And I believe there has been some work with using MSCs for lung issues because it's actually a pretty good delivery mechanism to get them to the lung. And otherwise, though, if the idea was to get them to some other target tissue, A, they're really not geared to become other tissues besides the mesodermal tissues. And B, it's pretty hard to get them there, at least if you try to do it through the systemic blood vessel flow. In contrast, the V cells at 1 to 2 microns, they're smaller than a red blood cell. So I've seen an image of a red cell and a V cell next to each other, and the V cells are about a quarter of the diameter of a red blood cell. And red cells make it through. So clearly, as a smaller cell, the V cells can get through, which does mean that they can be distributed throughout the entire body.
Mm.
There isn't hard evidence that the cells cross the blood-brain barrier, but some of the regenerative effects that we've seen really suggest that they probably do. The dual elegance of the method with the song laser and this whole line of work really began with the first experiment we did at the NIH. And when we built the first system, the waveform that we're using has a unique restructuring of how the waves relate to each other. And I might actually have to show the diagrams because it's a lot easier to understand. We created, in essence, a new form of laser energy by altering the phase relationships of how the waves stack up with each other. And before going into technically why it would be, the two attributes of it would be that instead of one continuous wave with all the waves lined up in phase, we create a pattern where the waves line up exactly out of phase, so they cancel the vectors. It's a vector sum zero. It becomes literally invisible. And energy in that form, based on the physics theories around it, would penetrate much more deeply through the body than ordinary photons. And then the photons that do remain are in a pattern of constructive and destructive interference, where most of the light becomes invisible and there are islands of constructive interference. And our physicists model that even the physical light packets can go 10 to 20 times deeper through tissue than ordinary laser light. So we had this new form of energy, and the question was, will it do anything? And it wasn't as though we had a brilliant hypothesis for what it should do or could do. We did an experiment with a colleague of mine where we took stem cell cells from the bone marrow, and we literally just passed the beam for different durations from as little as 5 minutes to as long as 60 minutes, and then put them back in the incubator and looked at them 24 hours later. And the remarkable finding, the reason we're doing what we are today, is that in every single flask from 5 minutes to 60 minutes, there was a visible line of cells where the beam had been. So we had stumbled upon or happened upon, or kind of like penicillin in a dish with Sandra Fleming, we made an observation that had implications. One of the biggest challenges in stem cell therapy is how do you get them to go where you want them to go and do what you want them to do? So what we had observed was what appeared to be an amazing way to give a type of directional localization signal, like a cellular GPS, to the stem cells to go to this location. And not just go to this location, but to actually adhere there, to stick to each other. And this we describe poetically as singing the song of the stem cells in light, literally kind of like a homing call with a Pied Piper of stem cells. But whatever metaphor you want to use, it kind of like a homing signal, actually, just a sort of a beacon saying where to go. And the fact that they were adhering to each other, the question was, well, what's actually happening at the biochemical level? So we looked at what is called the cell adhesion molecule system. And cell adhesion molecules are protein projections from the surface that allow the cells to stick to each other. The metaphor is cellular Velcro. And for that type of cell, the chemistry that was relevant was called the integrin system. It maintains the integrity of tissue. And the integrins are a dimer, which means two molecules together. It's an alpha chain and a beta chain. And we looked at alpha 4, beta 1, and beta 2 integrins. We did the stimulation with the song laser, and 24 hours later, we saw an up to 40 to 100% upregulation of the physical expression of the adhesion molecules.
Mm.
So we had a mechanism to explain not just that the cells appeared to be attracted to where the laser was, but that they could also stick together. And the reason cell-to-cell adhesion is important is that the cell adhesion molecules are not only responsible for the mechanics of holding the cells together in tissue, that uh, they are also involved in cell-to-cell communication. The integrin system is really pretty amazing. So there is an extracellular portion, which is the cellular Velcro part where they stick together. Then there's an intramembranous region that is also believed to have a trigger zone. As I recall, that particular amino acid sequence was GKFFR, or lysine, lysine, phenylalanine, phenylalanine, arginine, if you want to know that translation. And if that trigger zone is activated, it then sends a signal to the part of the protein that is intracellular, that is associated with transduction proteins, that the signal then gets transduced into a communication that then goes to the nucleus to instruct it to do things, in this case, to make more integrins. And when we talk about stem cells, the reason a stem cell knows that it should become a neuron in the brain and a heart muscle cell in the heart is that there are two main signals that tell the cell what to do. One of them is cell-to-cell communication, and the other is the microchemical environment around the cell. And I saw the work on the VSELs that depending upon the chemistry of the culture medium, that they could be, how you say, instructed to become nerves or pancreatic cells or muscle cells or cartilage cells. So those are the two mechanisms. The song laser then, with respect to the VSELs, we've done experiments to show that this is an efficient way with the type of signal that we're creating with the new type of wave pattern in these sparse nodes of constructive interference, these little islands of light, that also have very fast pulse repetition frequencies. And depending upon how the system is configured, the repetition frequencies can go as fast as sub-femtosecond. And you hear a lot about nanoseconds, which are a billionth of a second. And a femtosecond is even faster. A femtosecond is a millionth of a nanosecond.
Oh my goodness.
And molecular vibrations and rotations occur on the order of 10 femtoseconds. So the modulation frequencies can occur to give actual photoacoustic stimulation, creating a resonance signal, literally matching vibrational modes, that can go down to the level of single bonds between molecules, or larger groups of bonds, or parts of a molecule, or whole molecule. So the system is very elegant. And it really goes beyond just the song of the stem cells. The song laser system is really about singing the song of the molecules. And song itself, just like laser, is an acronym, which I learned, by the way, when I was 8 years old. I actually started with lasers in 3rd grade, which I could tell you then was light amplification by stimulated emission of radiation. That the song, besides being singing the song of molecules, is a Strachan Ovokaitys node generator. Scott Strachan was the brilliant physicist who developed this optical invention with me, and he was the one who coined the phrase sparse constructive nodes. So it's literally a song wave generator that is making nodes of visible wave packets that go deeper and can give very rapid signals. So the song has multiple meanings. And I like to acknowledge my co-inventor, because it was absolutely brilliant the way he figured it out. And he was the first scientist, when I told him what my ideas were and what I wanted to do, he's the first one that didn't say it was impossible. It was highly improbable, which mathematically is infinitely better than impossible. And in 6 weeks, he reduced the concept to a practical device. So pretty fun
Uh,
backstory on that.
yeah, that's
In our first,
amazing.
in our first experiment, literally what was in essence like throwing something against the wall and hoping it would stick. Um, if we hadn't made that observation, we probably wouldn't be talking today.
So I'd like to just recap a little bit here, okay? Just because there's been a lot thrown out there. So with the V cells, I've got a follow-up question here, but the V cells are injected, and the song laser is really the homing beacon that calls the V cells to a particular area, whether that be injury or an area that we want to address. That laser has a multifactorial component where now we're bringing the V cells to the area. It's releasing adhesion molecules. The cells are then lining up in that area. They're having communication both intracellularly or through the transmembrane, right, with each other. And then we're also changing the environment with the song laser to be able to promote new tissue growth or inflammation reduction of that particular type of tissue. We're getting translation into the nucleus to send different signals to be able to have that regeneration. So it's the combination of these two, the song laser and the V cells, that you've been studying and utilizing.
Right.
Yes. Okay, cool. The follow-up would be, one thing, where do we get the V cells from? We talked about umbilical. You've mentioned bone marrow as a potential location for mesenchymal stem cells. Where do the V cells, the autologous V cells that we would get from ourselves, where do we pull those from?
Mm-hmm. The beauty of it is that it doesn't require a little liposuction to get fat or putting a needle into a bone to get bone marrow. All that we need to do is start a single IV. The basic protocol draws 6 tubes of blood of 10 cc's each. Some people have that much drawn just for routine blood panel testing. And those are drawn in special gel separation tubes that by cell density, after 10 minutes, separates out almost all the red cells, typically 99%, and virtually all the white cells as well. And that leaves, at that point, what would generally be called PRP, or platelet-rich plasma, which contains plasma platelets, uh, growth factors in the plasma, particularly those from the platelets like PDGF, platelet-derived growth factor, but also the V cells, because the V cells are about the same size and density of the platelets. And we've only validated one tube, which is primarily designed for cosmetics. So it wants to make this very pure preparation. Red and white cells tend to be inflammatory. We inject someone's face with PRP. You don't want them to have more inflammation than just the inserting a needle will do. So it starts out basically as a very high quality white cell depleted PRP. And that then gets drawn into 2 syringes of about 20 mils each, of a person with a normal hemoglobin. You generally get about 40 mils of the plasma V cells and platelets from 60 cc's of blood. And so that's where it begins. So it's very simple. Because the V cells stay so young, the metaphor is almost like your cord blood equivalent is circulating in your body at room temperature. It doesn't need to be cryogenically frozen at minus 70 degrees centigrade, because they're metabolically dormant. We're not sure why they do that. They simply do. But it's useful for regenerative medicine because they stay so much younger biologically than the person. What was also fascinating in the work we did with my colleague, Dr. Peter Hollins, who I think Spun was part of the Nobel Prize winning team that discovered and developed in vitro fertilization, That was Sir Robert Edwards, his mentor, that got the prize, I believe, in 2010. So at his lab in Manchester, England, we did the study of identifying, well, what is the V-cell count in the preparation that's formed when you remove the red and white cells. And that number, strangely, has been all over the board. The count that we got was 800,000 cells per cc, which is a pretty reasonable number just from a blood draw. What was amazing was that immediately after applying the song laser to the syringes, that would be about 20 mils each, our typical procedure, that the flow cytometry to do the counts was repeated right after that was done. So there wasn't time for the cells to have much in the way of a particular change. The cell counts immediately increased to about 2 million per cc.
So we're getting
And
replication of these V cells.
well, we know cells don't divide that fast. And whether this is the way it actually works or not, we've published a theoretical biophysics paper. And our theory is that we are making the cells more countable. One of the main proteins that is used to distinguish a B cell from an adult differentiated cell is a protein marker called CXCR4. And CXCR4 will bind to a blocking molecule called EpiX4. And our biophysicist lead author on the publication did the mathematical modeling to suggest that we were using just the right amount of power and the right kind of power and frequency of stimulation to break the 7 salt bridges between EpiX4 and CXCR4 to liberate the protein that was blocking it, so that when the flow cytometry is done, and that works by using antibodies that will target a particular protein, that more of the antibody, which then is tagged to a fluorescent molecule, more antibody will be bound to the V cells that are present. So our theory is that they're not expanding the number, that we're simply through photoacoustic resonance dissociating a blocking molecule, make it easy for the counting antibodies to get there to make them more countable.
Wow. I find that very fascinating. As you mentioned, it couldn't be replication because it wouldn't occur that quickly, but the fact that you're able to free up the V cells to make them more countable, you're going from initially, you said like around 800,000 to 2 million. So is that when the blood draw occurs, the 6 tubes, it's spun down, the laser is initiated. Do you utilize the laser at that point when it's in the syringe, and
Yes.
then utilize the laser again for where you're wanting to turn on that homing beacon?
Right. So once the
Okay.
cells are activated with the laser, then the cells are infused IV, and we have what we call the laser guidance protocol, and that's individualized to the person. If you were to be able to magnify the regenerative effects in one part of the body or another, each person has their own unique characteristics of what the priorities are. And from what we've observed, it seems that the cells tend to find their new home in about 20 or 30 minutes. I'm sure some cells remain circulating for a while, but it seems like the greatest benefit of the laser guidance protocol to give a directional signal in a quantum sense to increase the probability that a stem cell goes in the volume of tissue where you most want it to go, then we do the laser guidance part. So it goes from the blood draw to separating the cells to the activation to infusion to guidance.
Got it. and which I think is really handling one of the primary difficulties that we had. We had multiple patients travel for stem cells when this was a hot topic, going to other countries. And, you made mention of it earlier, them circulating for about 6 weeks and not necessarily hanging around and creating the uh, potential regeneration that we were looking for, is that there was no way to home in where they were going. And often it would go to the area where the body needed it the most, whether it was an inflamed area. We had multiple patients come back after having this done, and they said they had improved vision for a period of time. But then over time, within a few months, it was back to needing their readers or their prescription glasses. And then the effects of it were very minor in the area that they were trying to target, whether that been a musculoskeletal injury, or the brain for cognitive dysfunction. So I find that very interesting. So with the laser guided, the song laser, we're not only activating the cells, but then we're able to guide them. So I think that handles a primary concern that I had in the early experience that I had with stem cells and patients being treated with them.
Right.
Yeah.
In essence, the laser supplies a signal sequence, go there, stay there, regenerate there.
So I think that can lead very nicely into what kind of results are you seeing when we are utilizing VSELs, or what kind of symptoms are we able to see improvement when utilizing VSELs?
It's fair to say when the body gets younger, things get better.
Yeah. So it's due to the fact that we're addressing aging through this process.
Mhm.
Yeah. Okay.
Yeah, and that would be probably our most interesting observation to date. And that has to do with looking at the metrics of biological aging. And I have a little clip with the co-founder of True Diagnostic. And
Yeah, we interviewed Ryan on this show.
yeah, so you know Ryan.
Yeah, very well. Yeah.
Yeah, he's a great guy.
Very good. Yeah.
And we did a 2-year study where we did a lot of their True Diagnostic True Age tests. And I don't know if you've gone into the different biological aging clocks yet and their relative accuracies.
We have, yeah.
Okay.
Yeah. Yeah.
So you already know that the most accurate clock is the one originally developed by Dr. Stephen Horvath of UCLA, also founded the Clock Foundation. He's all into biological age. That there is the epigenetic DNA methylation biological aging clock. If we already go into the details of that, I won't go into the technical parts, but it's a much more extensive survey of the methylation system and how that changes over time than what is probably better known, which is telomere testing. And of course, telomeres are ends of chromosomes that tend to get shorter each time the cell divides. So they get too short, they stop dividing, senescence occurs, and that's not what we like. So the methods of elongating telomeres are interesting. And according to Dr. Horvath, the telomere clock is only about 14% accurate. And the epigenetic clock that True Diagnostic uses and others in that space, though I particularly like how their work is evolving. And we're doing a cool project with them now that we can talk about because it's related to our FDA clinical trial that is in the works. And he basically said that when they first started looking at our results, they were shocked because they thought they were making a mistake. said they had to repeat the tests several times to make sure that the results were accurate. And ultimately, after repeated tests, they were certain of the accuracy. And he said that the results he was seeing were orders of magnitude better than any they'd seen for actually reversing biological age, not just slowing it, but reversing it. And one of the very interesting factors was testing people that had had the treatments, but we didn't have pre and post data. We just had, where they were after the treatments. And you've probably talked about the work of Dr. Greg Fahey with the study that used the combination of growth hormone, metformin, and DHEA.
Right.
And it was a big celebration when they showed 3 years of biological age reversal after 1 year, as well as some pretty interesting thymic regeneration. When he showed his data at 5 years, the 3 years of age reversal had basically regressed back up to their chronological age. So it wasn't a persistent effect. When we started testing people that had had a treatment anytime in the past 5 years, we found they were typically 3 years younger than their chronological age biologically. And it got more interesting when they had 2 treatments, because we were typically seeing about 6 years. And with 4 treatments, we even saw 12 years of biological.
Oh my goodness. That's crazy. So with the biological age test, just so everybody is on the same page, right? Biological age is looking at your cell, your body's age, your physiological age versus your chronological age, which is the number of years you've been around. And so we utilize this testing from True Diagnostics to get a gauge, and I'm very excited with where they are headed. They're doing a lot of cool things. But with this treatment, with the VSELs, we are seeing this biological age after having 1, 2, 4 treatments. What you're saying is we're seeing that the biological age is reversing and is maintaining that reversal, at least at this point, up to 5 years.
Right. Well, now it's long. There's more years since then, but those were our initial results. And then we were able to do pre and post treatment testing. And of course, it's more compelling. We did find that if people had not had one of our treatments, their biological age was usually the same or even a little older. So half a year, even a year and a half older biologically than the birth date age. And we then had the more scientific approach, because you couldn't be 100% sure that someone might have been biologically younger because of another reason, like great genetics or incredible lifestyle.
Sure.
And then it was still a consistent pattern. But we were seeing typically about 3 years of biological age reversal per treatment cumulatively. And in our group that were really interested in pushing the envelope of biological age reversal, where they did 2 to 3 treatments a year for 2 years, we have a group of people that's anywhere from 12 to 18 years younger biologically than their birthday age.
Gosh. Wow. I have not seen that result yet in practice. I think we're going to start doing the VSELs. That's pretty impressive. Well, I mean, that leads nicely into the other research that you're doing. And I know I'm pretty excited about, as I'm sure you are, about your FDA approval recently. So if there's anything that you can speak to on that, I'd love to hear a little bit more about that, where you're headed with that. And yeah.
I'm laughing because just at this hour, the window washers.
I know. I've seen them in the background there.
Really?
They're making those windows look younger. I'll tell you, they look good.
They're looking very spick and span right now.
That's great.
Well, there, of course, be a number of of clinical experiences we could talk about. And I think for this particular discussion, it's really good to focus on the biological age reversal. And as you can imagine, any system tends to get better if you provide young, robust, healthy cells, especially if they're your own. The most exciting thing that's happening, and I think is really landmark in understanding the deep biology of aging, is that we had the privilege of being invited to treat a baby with progeria. And for those that aren't acquainted with the disease, it's probably most popularly known as the Benjamin Button disease, because Brad Pitt, when he was born, looked like a typical appearance of progeria. What's amazing about this disease is that aging is accelerated about 10 times faster than normal. So a 7-year-old literally can look like they're 70 or 80 or 90. It's also incredible, and I muse upon things like this. There's been a lot of information activity around so-called SNPs, single nucleotide polymorphisms. And 23andMe really popularized this. And a SNP is the cute abbreviation. Single nucleotide polymorphism simply means that the DNA code is a triplet code. 3 bases in a row, whether it's an ACT or G, it defines what that particular part of the code will mean. So there's 64 different codons, 4 to the 3rd power is 64, and it's been fully defined. If you know what those 3 letters are, you know what it's going to be, which is either a start command, a stop command, or which amino acid to build to a string. And there are some nucleotides, some bases, if you change one letter to another letter, that will change the amino acid. And if that occurs at a critical position in an enzyme or a receptor or a functional protein, that one change in a critical position can significantly alter the function. So 23andMe was particularly about these minor genetic variations where a single base change results in an altered product that could have a significant impact, like the very famous MTHFR SNP, which has to do with the proper metabolism of folic acid. If it's abnormal, it tends to raise homocysteine, which has cardiovascular and other health consequences. It's actually pretty meaningful. Or a SNP that might mean that asparagus smells weird.
Mm-hmm.
So which might reduce your liking of asparagus, but doesn't really affect longevity or health. So what's amazing with progeria, that this incredible consequence of aging that is so fast, children die as young as age 6 from old age, average age of 12, is a single SNP. So if we're going to like put a weight on the most biologically harmful SNP versus almost no impact, the number one, as far as I know, would be progeria. It is 90% of the time caused by changing an A to a T at position 1824 on a particular sequence on the first chromosome. Changing that one base in 3 billion bases results in a cascade of consequences that the structural protein for the nuclear membrane called lamin A doesn't function properly. The nuclear membrane is unstable structurally and tends to bleb and bulge and collapse, which results in DNA damage accelerating 10 times faster than normal. That then translates to the consequences of progeria, where aging is accelerated in an extreme degree and resulting in very small children with, unusual physical features that tend to be intellectually normal, just have literally a very accelerated frame. So this disease is incredibly interesting because if we look at the model of extreme aging, and if we learn how to manage that, we'll probably have a better handle on the much easier issue of quote unquote normal aging. When I saw the baby, it was in a critical situation. I had been in discussion with the family for some time, working on getting IRB approval to do the treatment. And I'll explain what's interesting about the treatment. Because in this case, every cell of the body, in a sense, is not working well. The nuclei are all messed up. The cells divide slowly. Damage accumulates, and they tend to get skin issues, cardiovascular issues, digestive issues. So the use of the VSELs in this case is exceptional in that the allogeneic application is the strategy. And it turns out that at the time of the treatment, she was only 11 months old, which is great because if you can intervene with progeria before all the developmental challenges occur, there could potentially be a much more normal appearance and much longer lifespan. Mm-hmm. So it turned out that she has a 3-year-old brother. And the first thing we need to match if we're using allogeneic VSELs is the blood type, because the VSELs are believed to be the precursor of the hematopoietic stem cell that makes red blood cells. So you don't want to give mismatched VSELs to someone because you might literally create the type of blood that'll cause a reaction. Mm-hmm. That would not be good. So the brother was a perfect blood type match. But what was really remarkable is that the HLA testing showed he was a 90% match. They used a broader panel with 10 different antigens, and it was a 90% match, almost as perfect as an identical twin. It's known from bone marrow transplantation that you only need a 50% match for allogeneic VSELs. So we only needed 50%, and we had 90%. When we did the treatment, it was under a fairly extreme circumstance because the baby developed life-threatening, severe classic influenza A pneumonia that was proven by blood testing. The first night she was on 100% oxygen and was so hypoxic they almost put her on ECMO. It was that extreme. And she was a little better, but still on a fairly high percentage of oxygen when I saw her. So we did the treatment using her brother's perfectly healthy, normal, well-matched, blood type matched VSELs. And even within 24 hours, she was already better. The ICU doctors thought she would never get off the ventilator, and she recovered and was able to go home. She had very thickened skin. They initially thought she might have scleroderma. She was literally in a skin prison. And 2 weeks after the treatment, she could hold a toy and turn on her side for the first time. So on the strength of this case and the safety and logic of the rationale, probably the biggest breakthrough in our line of work for sure is that recently received an FDA acceptance to enter formal clinical trial testing. So it was literally an FDA acceptance letter to apply this protocol in a formal clinical trial for fast track FDA approval of a rare pediatric disease indication.
And so that's with the VSELs and then the use of the song laser.
Right, because the VSELs on their own may not be as effective unless they're actually biologically activated. And then we can also direct them to where that particular person is most affected with their um, clinical outplay of that genetic disorder. And we're going to be doing intensive epigenetic as well as biochemical testing. So we're working with True Diagnostic. I've had discussions with Ryan about this, that there's some things that are very interesting. Uh, uh, I don't have any precedent data on, for example, the so-called Dunedin pace of aging, which normally is one, and with our and other treatments, it can be slowed a lot more than that, which is, the desired goal of that. It might be as high as 10 in progeria. We don't really know.
Mhm.
There are also, as actually Hannah defined, they've already discovered certain epigenetic abnormalities that tend to occur in progeria. So we'll test for those, we'll test biological age, we'll test rate of aging. They now test the aging of various organs and systems. So we can look at a whole profile of epigenetic testing for looking at overall age and rate of aging, but also unique epigenetic attributes of progeria. A challenge is that the biologic age testing isn't really developed for infants and children, so they may need to make some algorithm adjustments to account for that. So it's very interesting from that standpoint. And then we're also working with Ginfinity. You probably know Jin Shangxi, you might have interviewed him too. And they have probably the most extensive biochemical panel related to aging chemistry as well. So we're going to look very extensively epigenetically and biochemically, and we will iterate that information into looking at various developmental characteristics for our clinical trial that we are now going through the steps of getting final approval of our ultimate protocol and going into what will be a 1-year clinical trial.
Well, it's such a win to see receive that FDA acceptance letter, as well as be able to help a very rare but very difficult condition in infants. And then I couldn't think of a better model to be looking at aging and longevity than progeria. So pretty, right? So it's pretty amazing what you're going to be able to, hopefully pull out from this study, and obviously really look forward to seeing what the results are. I'd like to close this interview, with, if people are interested in looking into VSELs or having the treatment, what's the best way to contact you or to get involved with the practitioner?
Mhm.
And then finally, just make a brief mention to the documentary that upon the release of this summit will have just been released, maybe a month before, the
Mhm.
Biohack Yourself.
Mhm. Yeah, thank you very much for that invitation.
Yeah.
We're sharing some of these stories actually on my Instagram, which I was inspired to do because of the movie that so many people are going to see. And that's at Dr. Todd O. Various interesting stories are being shared there, including speaking at the next World Changers Summit at the Vatican, which will be pretty cool. So that is one way just to follow. It's not efficient to contact that way. For information about what we do at our clinic, the website is simply qigenix.com, qigenix.com. And the email is info@qigenix.com. And you can just call the main number, which is 760-405-1908. So that's the basic contact information. Also, uh, you're aware that I have uh, patents on this and have licensed this methodology, technology, and protocols to a number of clinics around the country. There are about 3 dozen now. And for some people, it's not easy to travel. So if it isn't convenient to come to us, we can also match a specialty area of particular clinics and location and let people know what those options would be too if they call our main number.
Awesome. And this documentary that's coming out, Biohack Yourself, is being released. I don't actually remember the release date. Is the end of December or early January?
It's December 15th.
December
at the
15th.
4M.
Oh, that's right. At the A4M conference, right.
Yeah.
And then it'll be on what kind of what platform will people be able to watch it beyond the beyond
It's an interesting, yeah, it's a good question. So you'll be at the A4M. It's a red carpet event, December 15th. Maybe I'll see you there. It's going to be,
yeah.
It's being released on Amazon as the main channel, and then we'll probably go through Hulu and multiple other channels. As far as I know, almost any channel except Netflix, I think there's like a differentiated Netflix or other in that space. At least that's what I'm told. But primarily, it's going to come out on Amazon.
Awesome.
Yeah.
Well,
so
Dr.
Interesting.
Dr. Todd, I greatly appreciate your time today, the interview, your work, our friendship. Just to close out, first time I heard Dr. Todd speak was years ago at a conference from Stage. And blatantly I heard him and I'm like, this is hocus pocus. What are we talking about here? And then I saw him multiple times after that, and the evidence and the knowledge kept building and building and building. And the proof is in the pudding. I mean, the presentations that you've put on, the case studies that you've been willing to share have been miraculous. And so very honored to have you on here and be able to get this information out to more people, more ears.
Yeah, well, great pleasure. I like working with innovators like yourself that are pushing the envelope for creating very best outcomes. So honored to be with you. Thank you.
Amen. Amen, sir. All right. Well, thank you so much. Everyone have a great day, and we'll look forward to seeing you.
Thanks for listening to the Longevity Leaders Podcast. Our Longevity Leaders community is on mission to transform 100 million lives by 2040. If you enjoyed this show, please share this with somebody that you know, especially another doctor or physician, but also click that follow button. Until next time, stay strong and live long.